This tutorial is to describe the Queries and Tools page present on Apidb. This page can be accessed by following the link in the red navigation bar titled Queries and Tools. At the top of the page you'll see a listing for the two most frequently accessed queries, that is, genes by ID and genes by keyword. Next on the page, the queries are presented in a tabular format. These are broken down into various categories, the first of which are queries to identify genes by various parameters. These parameters include the genomic position of the gene, attributes of the gene, whether it's a tRNA, a coding gene, an rRNA, the intron-exon structure of the gene, queries to identify genes based on their expression, either transcript expression or protein expression, queries to identify genes based on their protein features and characteristics, such as the molecular weight and isoelectric point, queries to identify genes based on their putative function, as annotated by Go terms, EC numbers, metabolic pathway, yeast-to hybrid interactions, and also other predicted interactions. Queries to identify genes based on evolution, such as orthologs and paralogs, and various profiles. And queries to identify genes based on population biology, that is, what SNPs are present. There are also other queries to identify genes based on keywords, species, available reagents such as those from the MR4, queries to identify genes based on similarity or computed domains such as their protein motifs, blast similarity, and also queries to identify genes based on their cellular location. That is the gene a signal peptide, does it contain transmembrane domain, etc. There are also queries to identify genomic sequences by various parameters, such as the sequence ID, the species, and blast similarity, as well as queries to identify ESTs, such as by the extent of gene overlap, by the library present, by the blast similarity and location, queries to identify SNPs by various identifiers and their allele frequency and chromosomal location, and also queries to identify ORFs by the presence of mass spec evidence, by various motifs and chromosomal location. At the bottom of the page, there's a listing of various tools present in Apidb. There's the Sequence Retrieval Tool, or SRT, which allows one to query for sequences, and that can be of type of either genomic, transcript, CDS, or protein. There's a link to ApiPsych, which allows one to query for metabolic pathways present in all Apidb organisms. There's a link to be able to blast sequences against all Apicomplexus species, and a link to KEGG. In this query grid, you'll see that there are various icons, and this indicates the availability of data in the various API sites for this type of information. And A indicates Apidb, C for CryptoDB, P for PlasmoDB, and T for ToxoDB. If you'd like to see an overview of information about the query, just mouse over the query name and a window will pop up with a brief description of the query. One can click on this link to go to the query and see the parameters.